This is the mitochondria, where reactions and processes such as electron transport chain occurs. The mitochondria has two phospholipid bilayer membranes, the inner and the outer, with the intermembrane space in between them. The cristae are the folds formed by the inner membrane to increase surface area to volume ratio, to have more space to host the proteins and enzymes involved in cellular respiration. The inner membrane envelopes the matrix where the Krebs or citric acid cycle takes place. NADH produced from glycolysis in the link reaction in Krebs cycle, and FADH2 produced from the Krebs cycle are coenzymes that act like electron carriers. They are transported to help drive the electron transport chain.
In the first step of the electron transport chain, coenzyme NADH will bind to complex 1 and go through an oxidation reaction to produce NAD+, which goes back to the Krebs cycle, electrons, which go to the ETC, and hydrogen ions. The electron donated from NADH will energize complex 1 to pump the hydrogen ions against the concentration gradient, from a low concentration in the matrix to a higher concentration in the intermembrane space. This process creates an electrochemical gradient of H+ ions. Meanwhile, coenzyme FADH2 enters the ETC at complex 2 without going through complex 1. It undergoes a similar oxidation reaction, producing FAD+, electrons, and H+ ions. Since FADH2 didn't stop by the first complex, it's not able to help pump electrons in that step, and in the end, that results in fewer ATP molecules being made from FADH2. In the meantime, the electrons from NADH in complex 1 are transferred to coenzyme Q, and so are the electrons from FADH2 in complex 2. Coenzyme Q delivers both of these electrons to complex 3. At complex 3, the electrons energize the proton pump, and again, the H+ ions are pumped out of the matrix against the concentration gradient. The electrons are now transferred to cytochrome C, another mobile carrier which will carry the electrons to complex 4. At complex 4, the electrons from the electron carriers will also energize this complex so it can pump protons from a lower concentration to a higher concentration in the intermembrane space.
The movement of electrons passing through each complex to energize them will continue to where the electrons combine with the oxygen from when we breathe in, the final step of the electron acceptor, and H+ to produce water, or H2O, which is the waste product of ETC, and the final step of cellular respiration. The electronegativity of the O2 is what keeps the electrons moving throughout the ETC and towards the oxygen. Without oxygen, this process cannot occur, effectively halting the entire ETC. This is the only process that is aerobic, which means it includes oxygen.
These processes of pumping protons out of the matrix and into the intermembrane space lowers the pH, contributing to the growing electrochemical gradient.
This leads to chemiosmosis, where the proton concentration gradient powers ATP synthase, a protein embedded in the inner membrane of the mitochondria to produce ATP.
Because the nonpolar tail of the phospholipid bilayer of the inner membrane rejects any charged molecules, H+ can only enter the matrix through ATP synthase. As protons pass through the ATP synthase, the enzyme will rotate, which produces mechanical energy that changes the shape of the active site on ATP synthase, enabling the enzyme to catalyze the conversion of ADP and inorganic phosphate into ATP. This is called phosphorylation, the process that is responsible for the bulk of ATP production during cellular respiration, and the whole ETC process is called oxidative phosphorylation.
This process of chemiosmosis and the creation of an electrochemical gradient to make ATP is the primary purpose of the ETC.
To summarize, in the ETC process, the reactants are NADH, FADH2, O2, and ADP plus inorganic phosphate, and the products are NAD plus, FAD plus, water, and ATP. Overall, oxidative phosphorylation via the ETC produces about 34 ATP molecules, while the substrate-level phosphorylation during glycolysis and Krebs cycle produces only 4 ATP molecules. This adds up to a net total of 38 ATP per oxidative glucose molecule, showing how oxidative phosphorylation is a much more effective way of producing ATP, which allows many of the body's systems to run.